is table of content. So we have objective, introduction of rotifer, introduction of copepod, introduction of moina, method zooplankton sampling, method identification and data collection, method data analysis, hypothesis statements, result on hypothesis, conclusion, challenges, future direction of the study, implication of the study, summary, and references.
The objective of this experiment, the first one is to sample and identify rotifer, copepod, and moina in ponds and lake at UiTM Muka Sarawak. The second one is to isolate and cultivate identified zooplankton species. The third one is to evaluate the growth and population of zooplankton fed with fish meal. And the last one is to analyze data using ANOVA for statistical comparisons.
First, I will explain about rotifer. So what we know about rotifer? Rotifers are small freshwater and marine invertebrates. They mostly eat algae, bacteria, and dead plants and animals. Rotifera animals are called for their whale-like ciliate corona at the front end, which they use to move and consume. Rotifers are attractive aquaculture food because they are tiny, simple to cultivate, and nutrient-rich.
The next is introduction of copepod. Copepod are tiny crustaceans which are normally found in marine and freshwater environments. They play a crucial role in aquatic food chain as first-order consumers and as a high-quality source of protein for fish larvae. Due to their high nutritional value, copepod have used in aquaculture as live feed.
Next is introduction moina. Moina are small freshwater crustaceans from the Daphnidae family, widely used in aquaculture as live feed due to their high nutritional value and rapid reproduction. Ali et al. 2010. Under ideal consumptants, their primary method of reproduction, parthenogenesis, allows for rapid population increase. Nandini and Sarma 2000. By consuming algae and suspended organic materials, moina also contribute to the improvement of water quality. Benzi 2005.
The first method is zooplankton sampling. The material used for this sampling is beaker for sample collection and temporary storage, plankton net to filter zooplankton, and lastly, plastic pipette to transfer zooplankton samples. The next one is preparation of zooplankton sampling. Step 1. Collect the water sample of your ATM Mukah Lake using a beaker. Step 2. Filter the water using a plankton net to capture zooplankton. And step 3. Took the sample to the lab to observe it under a microscope. Step 4. Use a microscope to see and count the zooplankton. Step 5. Each species were recorded in Excel. As you can see in the picture, these are the example of zooplankton that we have collected from the water in UiTM Mukah Lake.
Next method is identification and data collection. So step 1, observe collected samples under a light microscope and identify rotifer, copepod, and moina species. Step 2, isolate species using a fine plastic pipette and transfer them into individual culture containers. Step 3, measure water quality parameters such as temperature, pH, and turbidity using a digital meters or test kit. Step 4, monitor and count the population growth weekly using a consistent volume sample. Record all data into a structured Excel spreadsheet, including species type, population count, week, and water quality reading. So, we use complete random design because each experimental unit received only one treatment and the conditions were uniform. The CRD is appropriate here due to ease of implementation and homogeneity in lab setting, same like temperature and feeding schedule. So, temperature, pH, and turbidity were measured using a pH meter and readings were taken at the start of the experiment. So here is how we put the data and we put in the Excel. It's week by week.
Next, we go on the methods for data analysis. So, at the table is our data collection from sample 1 until sample 6 by the week for rotifer, copepod, and moina. So, we use ANOVA single factor to calculate the p-value on this data collection. The reason why we use ANOVA single factor because it helps to determine whether there are statistically significant differences in the mean population densities of zooplankton such as rotifers, copepod, and moina across the different sampling locations in the lake. So for our data collection, the p-value are less than 0.05. So we accept the null hypothesis and reject the alternative hypothesis. Next, for the hypothesis statement, the null hypothesis, there is no significant difference in zooplankton population distribution in UiTM Mukah Lake. Next, alternative hypothesis, there is a significant difference in zooplankton population distribution in UiTM Mukah Lake.
Next one is result on hypothesis rejection or exception. As you can see in the method of data analysis, we perform a one-way ANOVA test to compare the population of rotifer, copepod, and moina in UiTM Mukah Lake. The results show a p-value of 0.00044, which is less than 0.05. That means there are a statistically significant variation in the zooplankton population. Therefore, we accept the null hypothesis and reject the alternative hypothesis.
So, for the conclusion, among the three groups, copepod has the highest average population, which is 56.67, and rotifer followed with the average of 43.33, and moina shows zero population in all samples. So, this result demonstrates that copepod dominate the zooplankton community, likely to their reverse adaptability and reproductive capacity under the existing environmental condition in UiTM Mukah. Rotifer also shows a relatively stable presence, while the absence of moina could be due to lower tolerance to prevailing water quality or ecological competition. These results are in line with earlier studies such the one conducted by Dumps et al. 2011, which found that copepod predominates in tropical freshwater system because of their rapid rates of development and resilience to environmental stress. In a similar vein, Seegers 2007 highlight the importance of rotifer as colonizers in freshwater environments, despite the fact that copepod frequently outcompete them in nutrient-rich environments. So, next for the challenges. So, first is sampling depth control. Inconsistent plankton net depth across site may have affected species composition in the samples. Next, microscope fatigue. Counting hundreds of micro-sized organisms manually can cause eye strain and slow progress. Next is moina absence. Moina was not detected in any of the samples, possibly due to unsuitable water condition, poor sampling timing, or natural absence in the site.
The next one is the future direction of the study. In the future, we will have the ability to gather data during various times of the year so we can observe what the season do to zooplankton population. The experiment can also be conducted in other ponds in UiTM Mukah to verify if other water bodies have a different count of zooplankton. Then, we can include additional water quality tests like dissolved oxygen or nitrates to see how they will affect population size. Statistical technique can be utilized to gain a better understanding of water condition as well as zooplankton interaction.
Next, for the implication of the study, first is biodiversity data. So, it provides essential baseline information on the diversity, abundance, and composition of zooplankton species and local aquatic ecosystem. Second, aquaculture potential. So, aquaculture potential, it identifies potential live feed species such as rotifers, copepod, and moina that are suitable for larva fish production in hatcheries. It supports the development of sustainable aquaculture practice through the use of locally available zooplankton. Next, water quality indicators. Water quality indicators act as a biological indicator for water quality since zooplankton population are sensitive to environmental change, and it helps to detect early signs of pollution or ecological imbalance in pond and lake ecosystem. Other than that, local cultivation. So what we know about local cultivation? Okay, local cultivation, it demonstrates the feasibility of isolating and cultivating native zooplankton species for commercial or research purpose. And it encourages cost-effective live feed production by using locally sourced organisms to reducing dependency on imported alternatives. The last one is scientific contribution. Scientific contribution can enhance the understanding of zooplankton ecology in tropical freshwater system, and it provides valuable data that can be used in future research on fish nutrition, feed formulation, and aquatic biodiversity conservation. The last one is summary. The group project met all objectives successfully. The findings offer significant insights into the ecological health of UiTM Mocha lakes and establish a foundation for future ecological and aquaculture research.
So, these are the references that we all use in these videos.